Okay, so the first module we'd like to work on today is pathobiology of asthma. So I like this definition of asthma. It's from Saul Perman, who's a famous physician scientist. He says, asthma is like love. Everyone knows it, but no one can agree on the definition. Well, what we know about asthma, I'm just going to move the pictures over a little bit. There we go. Is that it's a chronic inflammatory disease of the airways, the bronchial tubes, and the tubes seem to more easily constrict too easily to a variety of environmental stimuli. So there was a group called EPR3, Expert Panel 3, Recommendations 3, to develop a consensus definition of asthma. And we've gone all the way from love to this mouthful of definition about asthma, but it's very specific. So asthma is a chronic inflammatory disease of the airways in which many cells and cellular elements play a role, in particular mast cells, eosinophils, T lymphocytes, macrophages, neutrophils, epithelial cells. In susceptible individuals, this inflammation causes recurrent episodes of wheezing, breathlessness, chest tightness, and coughing, particularly at night or in the early morning. These episodes, the definition goes on, these episodes are usually associated with widespread but variable airflow obstruction that often is reversible either spontaneously or with treatment. And the inflammation also causes an associated increase in the existing bronchial hyperresponsiveness to a variety of stimuli. Reversibility of airflow limitation may be incomplete in some patients with asthma. So that's a mouthful, but you can see that it's hard to actually put your hands around what the exact definition of asthma is. But we'll be going into some detail about what we know about the underlying pathobiology, how we, in practical terms, define and assess asthma and its control, and some treatment considerations in the later parts of the talk. So asthma has an enormous disease burden, affects all races and ethnicities. In the US, it's estimated that 8% of American adults, 10% of American children have asthma at any one time. And globally, 300 million people are afflicted with asthma. It doesn't pick a certain age group. Anywhere from infancy to old age can be affected. Early in life, boys more so than girls, but after puberty, women more than men. And there's been a very large increase in the prevalence of asthma over the last few decades. Here's the map of asthma, and it's somewhat incomplete. You can see it's from 2004. The green areas are the reported highest areas of prevalence of asthma. So you can see in particular in North and South America, Australia, parts of Europe, you can see the red and brown areas. And you can see that for much of Africa, we really actually don't know. There's no real standardized data available. But providers, and I'll be interested in the question section later, tell us that asthma is quite prevalent in Africa as well. So who gets asthma? What are the risk factors? Well, most often it's children. Children with an allergic predisposition and who are exposed to what they're allergic to, so the allergic exposures. In the adults, we see this with occupational exposures. Sometimes this is called occupational asthma. There is a gene by environment interaction that leads to the clinical expression of asthma. So in this slide, I'm talking about the genetics of asthma, and we'll come to the environment in just a moment. But in terms of the genetics, there are a few summative statements, I would say. The first is that there are several genes called single nucleotide polymorphisms. This is a measure of genetic variation. So there are SNPs in several genes with asthma. So this raises this point that we'll come back to time and again, that asthma is more of a syndrome than a specific disease. It's very heterogeneous in terms of its genetics. There are a few of these SNPs, these single nucleotide polymorphisms, that are associated with all ages, and they seem to be related to airway epithelial damage and activation of inflammation. Interestingly, the genetic loci that are very strongly associated with IgE antibody levels are not significantly associated with asthma, with one exception. This is from a New England Journal of Medicine paper in 2010, and it shows what's called a Manhattan plot. To see if I can move this a little bit. Okay, so the Manhattan plot shows positive hits with a p-value. This is a statistical level that are high enough, this is the whole genome, that to be associated with asthma. And there are some genes here that were somewhat expected. For example, interleukin 13, which is an important part of type 2 inflammation. But interestingly, some novel genes came up in this screening, and this one in particular is the study of intense research right now. Okay. This shows in a table, in summary, IgE levels and the genetics associated with that, and here are the genetics associated with asthma, the chromosome where the variation is, and the gene targets. And the most important thing to show you here is that the gene targets for asthma are extremely different than the gene targets for allergy more generally, IgE levels, with this one exception of the IL-13 gene being seen in both. And there's development underway right now to use an antibody, to develop an antibody as a therapy against IL-13. Well, here's a graph for the age of onset for asthma, and this is showing that you can see the incidence rates for asthma are highest in the lowest age group, so children, infants really, but certainly by ages 3 or 4. So a good bit of asthma is developed already in early life. But you can see that even individuals older than 50 can have an incidence, a new onset of asthma, both males and females. Regarding the environment, there's some protective factors that have been identified, and these make it a little less likely to get asthma. So gene by environment interactions have been identified for smoking, for viral infection in early life, for siblings, and for farm exposure. Let's take a moment to look at the farm exposure information. So here is from a European cattle farm, what life is like in the barn. And there are 2 studies that were done that were published together in the New England Journal of Medicine a few years ago that shed light on this. One was called Parsifal, the other was called Gabriella. Parsifal looked at bacterial DNA in the mattress dust in the homes. Gabriella looked at children's room dust for both bacteria and fungus. And what they saw, this is probability, and this is basically the amount of the microbe. And in Parsifal, you can see the amount of microbes increase based on living on a farm. And then this is the incidence in red of the probability of having asthma. And you can see the more bacteria in the mattress, the less asthma. And this is, I'm not sure if you can see this fully. The images are blocking it on my screen. But this shows in blue the amount of detectable bacteria and fungus. And there's this inverse relationship to asthma. So living on a farm increases exposure to microorganisms, but it also protects because it's decreasing atopy, and shown here, asthma. Well, schematically, this is the key features of asthma. Symptoms result from some combination of airway smooth muscle constriction and bronchial inflammation. And the symptoms are cough, dyspnea, chest tightness, and wheeze. And you can see the narrowing of the airway that can occur in the asthmatic airway compared to the normal airway, the normal bronchiole. You can see mucus in here, edema, and basically the airway is constricted and narrowed by the inflammation and mucus. This is what it looks like under the microscope. This is a patient who unfortunately died with status asthmaticus here at Brigham and Women's Hospital. And here you can see the middle of the airway, there's a mucus plug. You can see the mucus metaplasia, of the airway epithelial cells, lots of goblet cells with mucus. You can see all these inflammatory cells. You can see the proliferation and hypertrophy of the bronchial smooth muscle, and all of this together constricts and narrows the airway. People can't breathe, and unfortunately in this case, suffocated. So there's mucosal, submucosal, and adventitial disease. There's excess mass of smooth muscle, as I showed you, and the contractility is abnormal. It's twitchy airways. There are many examples of cells that infiltrate here: eosinophils, lymphocytes, and mast cells. There's excess collagen. Here's the basement membrane. You can see it's like a magic marker thick. It should be razor thin. So there's been deposition of collagen, and this causes airway remodeling. It's abnormal. Here are the inflammatory cells. Some of them you can see are red. This is the eosinophils. And then the others are lymphocytes and even neutrophils. The smooth muscle layer is down here. Here's the airway lumen. So here's the airway epithelial cells. Here is some evidence for secretions. There's mucus, it's called Kirschmann spirals, and desquamated, sloughed epithelial cells that are called Crayola bodies. From the earliest days of the invention of the microscope, people have been looking at the secretions of asthmatics, trying to figure out what's going on. And so early pathologists described these expectorated secretions. So here's the T cell response. So this is the adaptive immune response in asthma. And there's Th1 and Th2 in this review by Bill Bussey that in early 2000s in New England Journal. Factors that would favor Th1 would be having older siblings around, spreading germs, early exposure to daycare, some infections like tuberculosis, measles, hepatitis A, and living in a rural environment, being exposed to microbes. So that favors Th1 phenotype, the skewing of the immune system, and this creates more of a protective immunity against infections, but also against the allergies. But factors favoring a Th2 phenotype, which is associated with allergic diseases, including asthma, widespread use of antibiotics, Western lifestyles, an urban environment, there are some dietary factors, and sensitization to indoor allergens like house dust mites and cockroach. This is overly simplified, but because there are some other cells that have been discovered, like regulatory T cells, Th17 cells, and innate lymphoid cells, but it does convey nicely this issue of balance in the immune response, and that there are factors that can favor Th1, which limit risk for asthma. This is a schematic showing mast cells and mast cell interactions with smooth muscle cells. So here, IgE antibodies, these antibodies of allergy, engage specific receptors. The mast cells, by detecting the presence of the antibody, release mediators into the environment, and these can activate the B cells to cause more IgE production. And you get a whole cycle of a positive cycle of allergic type inflammation. When the IgE levels are very high, you can see that this is amplified further, and this can be very prominent via IL-4 and IL-13, activating further production of IgE by B cells. So there are now drug targeting efforts going on to target IL-4 and IL-13 to try to block this cycle. This is histology from an airway of an asthmatic, and these red dots here are all mast cells that are embedded this is higher magnification, embedded in the smooth muscles. So if the mast cells release mediators that can act on smooth muscle, it causes this increased twitchiness of the airways, this excitability of the airways, and the stronger constriction of the airways. One of the types of mediators, one of the families of mediators that mast cells produce that can act on smooth muscle are called leukotrienes. These are products of arachidonic acid metabolism, and you can see that there's an enzymatic pathway to form leukotrienes, here B4 and leukotriene C4. These leave the cell of origin, so the mast cell, for example, and they can act on neighboring cells to cause leukocyte chemoattraction. And here are the cysteinyl leukotrienes, C4, D4, and E4, And these can engage this receptor, the cystinyl leukotriene 1 receptor, cis-LT1. And these are present prominently in smooth muscle and other places in the airway. And if these leukotrienes engage this receptor, you get smooth muscle constriction, eosinophil migration and activation, and edema formation. It was the biochemistry and biology that was recognized in these pathways that led to the development of specific anti-asthma drugs that are anti-leukotrienes. Xyluton can prevent arachidonic acid metabolism to leukotrienes by inhibiting 5-lipoxygenase. And these drugs are all receptor-level antagonists of cis-LT1. In the US, montelukast is prescribed quite frequently for asthma. Well, the airway epithelial cells interacts also with structural cells in the lung, these fibroblasts, sometimes called a mesenchymal trophic unit. And this appears to be disordered. There are barrier defects, so there's more permeability of things that we would breathe in that can actually get past the epithelial cells into the structure of our lungs. There's failed repair responses to injury or inflammation, and there's impaired antiviral host defense. So along these lines, viruses, especially rhinovirus and human rhinovirus C in particular, are linked to asthma exacerbations in children and adults. And it turns out that our antiviral protection mechanisms in the airway are related to the production of interferons. So one of the types of interferons is shown here from this publication. And you can see this relationship to change in lung function, FEV1. Healthy is shown here in blue, and in red are individuals with asthma. And so you can see there's a relationship in the ability to generate interferon to lung function. And so it's just this notion that asthmatics seem to be impaired in their generation of interferons that make them susceptible to viral infections and exacerbations. All of this leads to airway remodeling. I showed you some of this on the histology slide, but you get the mucus metaplasia, you get the smooth muscle hypertrophy and deposition of a lot more matrix like in the basement membrane. This is what a Crayola body looks like under the microscope. In expectorated sputum, you can see some inflammatory cells here in the mucus as well, but these are airway epithelial cells that have been sloughed off the lining of the lung. This is what a Kirschmann spiral looks like. It's really pretty, actually. These are mucus plugging of some of the small airways. You can see all the tortuosity here. And what you see is it's a coiled sheath of mucus, and these small cells that are lining it are airway epithelial cells. So you can see why this drew the attraction of somebody looking through the microscope, seeing these beautiful structures. Well, there's been a lot of work on the immunology of asthma, and the science is continuing to expand. But there are many pathways and mediators that together can cause what we clinically recognize as asthma. So I've talked to you about antibodies. There's cytokine mediators and chemokines and peptides. There's something called chitin-like proteins that can do this. I showed you leukotrienes, and even gases like nitric oxide can do this. And this is some of the cellular elements of the allergic immunology response. And now there's a lot of drug development efforts trying to block or change the signaling for each of these molecules that I've shown here. So that's the pathobiology section of the talk.